Our next Gilbreth lecturer is Dr. Ishi Keenum. Dr. Keenum. Dr. Keenum is an assistant professor in environmental engineering at Michigan Technology University. She completed her postdoctoral work at the National Institutes of Standards and Technology, we call NIST, in the Complex Microbiological Systems Group, and her PhD in environmental engineering at Virginia Tech University. Ishi will address innovative engineering strategy tackling the global threat of antibiotic resistance through a One Health framework. Please welcome Dr. Keenum.
All right, hi everyone. I'm really excited to be here. So I have a question for you all. How many of you all have ever taken an antibiotic? Okay, and it worked, right? You're still here. This is great. We want these things to stay effective. These are really important medications that almost all of us have had at some point in our life. So today I'm going to talk about, I'm an environmental engineer, and environmental engineers do a lot with how we make safe drinking water, and also how we can safely treat our wastewater. So here, I like to start with a picture. This is the treatment plant that's about a mile from my work. And you can see we're on this lovely surface water body. I don't think I have a laser. I do, okay. So we're on this lovely surface water, that's Portage Lake, and we discharge. And we have to think about what did we create wastewater treatment plants to remove? And most wastewater treatment plants were designed to remove fecal bacteria, which is great, because if you've ever heard of Oregon Trail, no one wants to die of cholera. It's bad news. But today we're gonna talk about antibiotic resistance specifically. And I think it really helps for us to understand what is antibiotic or antimicrobial resistance, and why is it such a problem? And then how does it evolve? What's going on here? So if you think about a microbial community, so microbes are everywhere. They are on the surfaces you're touching, and they're different everywhere. So if we think about a microbial community, we might have, some little red guys, we got these little long rod shapes, we have some green ones, we have this dark green one who's a little bit different, but he's kind of similar to those green ones. We got some cool brown ones. And so if you get sick, you might take an antibiotic. And so specifically, maybe your antibiotic is really gonna target those green guys, right? It's gonna take them all out. So goodbye, little greens. But remember, we had this one who was actually related to the green ones, but was a little bit genetically different. And actually that one was able to survive. And so we had him here, and then actually it might keep growing, right? So it survived this big threshold, this population, we were trying to get rid of it, and now all of the ones that kind of filled that niche actually are resistant to that antibiotic, or that way that we want to treat this disease. And this can be really problematic, right? Because like I all said, antibiotics are super important. They keep us healthy. They're one of the main ways that we have to get rid of bacteria that are causing infections. And I'm an environmental engineer, so why am I talking about this? It's really because antibiotics and antimicrobials are used outside of just human medicine. I think us as humans have probably interacted the most with the human medicine side, but we have to remember that things like animal agriculture, so this could be, we raise cattle, or we're raising chickens or pigs or aquaculture. Those all will use antibiotics for different purposes, and it's not always bad, right? Historically, we were using antibiotics for growth promotion. We've ended that practice, but we still, if an animal gets infected, like a cow or a dairy cow, and it has mastitis, we'll still treat that infection. And cows outnumber people in the US by about 100 to 1. So it's a much larger scale. We also can think about companion animals. So those are like your cuddly, your dog or your cat. They actually can get a lot of the same antibiotics that humans can get. And so this can cause potentially an interaction. We also have to think about where we waste streams. So when we make antibiotics with pharmaceutical companies, the places we make those, we have to make sure those will sometimes get into the environment, especially if they're in countries with not as stringent effluent regulations. And all of these things kind of come together where we start to think about how is this going to impact our broader environment? And they're all connected, right? They're not just happening in silos. So like the runoff water from where those cows and those pigs graze might come into your environment. And specifically, what we're gonna think about is this irrigation piece. But I'm not the one who came up with this. I do not wanna take credit. People much smarter than I figured this out. The World Health Organization is really thinking about this from a One Health perspective. And this is the idea that all of our systems are connected, and that in order to have a healthy planet and to have healthy systems, we need to be thinking about kind of all of our systems as connected and how we're gonna kind of move forward with it. And so kind of one of the questions that comes out of this is, okay, so this is a problem. It's a problem everywhere. That's great. How are we gonna measure it? How do we track that this is a problem? And conventionally, we really relied on clinical samples, and that's kind of the only way, but that really only targets that human piece. So how do we measure antibiotics or antibiotic resistance? The first way, the more traditional way we have is culturing. So culturing, you guys might have heard about in biology, but that's where you take a living organism, either from your environment or from a swab, and you grow it up. You have media that just selects for that organism. And then you can sequence it. So we use sequencing as a way to get the genetic sequence of what is that organism made up of, and then we can annotate to existing databases. However, in the environment, most of our culturing methods were really developed for clinical applications, so we're not great at targeting organisms that we're interested in the environment. And so what we can do as well is we can do direct DNA and RNA extraction. So you just take all that, you basically take all those microbes, you grind them up with beads. You can't see the bead, everything's very small, unfortunately. But then you get their DNA, and then you can directly sequence it. So this is what we're gonna talk about today. And we're specifically gonna talk about kind of the context of water reuse. And I know in California, this is incredibly relevant. There is a global water shortage, and I think Southern California feels it more than where I am at in Michigan. But when we think about other ways of using our water, so if we wanna take our wastewater and we wanna use it for other applications like irrigating, food that we're gonna eat or food that other animals are gonna eat, or in the case of direct potable reuse, water that someone else might drink. We have to really think about our systems removing antibiotic-resistant bacteria, even though recognizing they're not really designed for that purpose. So the data I'm gonna present, this was a study we conducted a few years ago now. We sampled a water treatment plant that was doing water reuse just for irrigation. So this is called non-potable water reuse, so don't drink it. And what this looks like is we have a treatment facility. So wastewater treatment is really multi-barrier approaches. It is made up of, we've got our primary, we've got all these different barriers that we use to remove bacteria as they go through the network. And we cultured it 4 times throughout the year, and we cultured and we did sequencing. So we did both of those methods. And we started by just measuring, so E. coli and Enterococcus, those are the fecal pathogens that we are regulated on as wastewater utilities. You better meet those regulations. But we started just to look at, looking at the total amount, which is what the regulation is on, and the resistant amount. Do we see anything? And we see, as we proceed through treatment for this reuse train, we start out pretty high. So these are on a log scale. So think of everything like 10 to the 8 or 10 to the 9. Raw wastewater is disgusting. So there's lots of bacteria in there. And then we have about 1% of our population is resistant. But we do see as we go through treatment, while these numbers both go down, and that's what we would expect, we see that proportion of resistant bacteria actually gets closer to the total bacteria. So those resistant bacteria are somehow surviving disproportionately to the total amount. We also see that we go below the detection limit. We see a little bit in the distribution system, but not a ton. If we look at Enterococcus, this is it's the same treatment facility and the same plot setup, but we see a similar story. We get this really great log removal through the treatment facility, which is what we would expect, and we always regulate kind of at this chlorination point. But what we see is actually when you start to wait in the distribution system and you go maybe at a short distribution point or a long distribution point, that we actually get quite a lot of recovery, specifically of Enterococcus. So this is culturing. So this is a live organism. We're not really We have its genome, and it's actually, we can see the resistant and total populations are actually fairly identical. So it looks like we actually are kind of enriching for these bacteria. And so then I wanted to see, okay, so this is just 2 organisms, right? E. coli and Enterococcus. There are about 2,000 at least different species of bacteria in even the human gut, but also in your wastewater system, because they all are very connected. So is there any way we can look at all of the genes that are present all at once, not just targeting the bacteria? Now, I do want to present the caveat that this means you don't know if the organism is alive or dead, and that's a really big limitation. So what we're going to see here, it's going to look like stacked bar plots, but you'll also see there's going to be points over it. That's just telling you the total amount of bacteria that are through your facility. So what we see is what we would expect, right? The total amount of bacteria is decreasing. This is really important because my take-home is wastewater treatment is effective. It is removing 10 to the 6 or 10 to the 8 of the bacteria that are going in. We don't need to overhaul the process. We just need to figure out new treatment strategies to target specific ones. But what we do see when we look at specifically resistance genes, and on this side, what we've colored them is by the drug that they convey resistance to, that are influent in our initial stages of treatment are significantly higher. They come in kind of at this stage, and it looks like primary treatment doesn't really remove them. But once we get to activated sludge, which is a biological process where we enrich for specific microorganisms, we see that we get significant decreases in resistant bacteria, and that maintains really until we get to chlorination. It starts to get, honestly, the error bars get bigger, but what we are really interested in is actually the color that makes up the bars. So what is present is really changing, and we think about that like profiles. And then we see in the distribution system, we had to break the axis because the graph looked terrible if we didn't do that. But we see significant increases with the secondary method for these genes through the treatment facility. So now we have 2 methods that are measured separately. I'm a methods person, I can talk about methods forever, but they're telling us the same thing, that we have recovery in the distribution system that we need to look at, especially if we think about risk. Like, is this gonna convey a bigger risk to people at the end use? We can also look at this, so those bar graphs are great, except for it's hard to see how those different components are actually changing because they're just like colors and they're all smushed. So we split those out where we just start to look at the difference in our later stages. So we think of this as just in the water reuse part, and we just track the different drug categories. Because all resistance genes are not created equal. Some of them are more clinically relevant, some of them are more native in different bacterial populations, and some, beta-lactam specifically, is one that we watch a lot because that's where a lot of our last resort antibiotics come from. So if nothing else is working, we throw that at it. But we can see if there's certain drug classes that are actually recovering more than others, and that we are still getting removal across some of them. But the take home is really that our non-potable water is containing resistant bacteria. I wouldn't say that's remarkably alarming, but it really means in terms of communication for risk that we need to be talking to people about how are they using their reclaimed water. This sampling was done in Florida. And so in Florida, they have the purple pipe system, but you can use that water for residential irrigation. And when you think about, at least when I was a kid, I loved to play in sprinklers. That presents a potential risk to an at-risk population, right? That's gonna have a different immune system characterization. So really, communication is key, but we also need to understand we don't have safety limits right now for what is a reasonable risk, and we need to move that forward. And so I was a graduate student when I started some of this work, and this really took me on a journey. My real question was, okay, so we saw this thing. How does this compare to what other people saw? Is anyone else seeing the same thing as us? And one of the real limitations we came to is that these workflows, these sequencing workflows, so culturing is fairly reproducible. However, you only get those 2 data points. It's really limited because it's so time consuming. Our workflows are really reproducible for one lab in one place. So you get those tight error bars, but they're not very comparable. And it's really because when you do one of these workflows, I presented it as kind of a, oh yeah, you just extract the DNA and it's magic, you get these sequencing results. It represents a lot of decisions that we have to make through our treatment, through our processes in the lab as scientists. Like, how did we collect our sample? How did we choose our sampling? How did we extract that DNA? Which kit did you buy? There's a lot of different ways you can do this. And then when you do your sequencing, what platform did you use? How many reads did you throw at it? We have all these decisions. And really, this starts to affect our ability to compare our data. And this is kind of why I went to National Institute of Standards and Technology, because it started to really bother me. How are we gonna do this better? And so some of the work that's come out of that group is we sent the same sample to 17 different labs. So we sent them a homogeneous fecal sample, and we said, you analyze it however you want, just send us back your data. And we're back on the log scale here. But we see that we're getting about, this is a 100-fold variation in what different labs are reporting. So this is really calling to question, and at NIST, we were thinking a lot more about the fecal microbiome and diagnostic for fecal microbiomes, where people are sending their sample off, and they're being told medical advice when they get it back. So while I started with the environmental engineering approach with those challenges, honestly, I've come to this being a much bigger problem when we think about healthcare and these diagnostic decisions. Because some of these were commercial labs, and if every time you send your sample, you get a very different result, not because there's a ground truth change, but because of the decisions that they made, it could make you think, oh, I did it, maybe I started to eat better, I'm eating more oatmeal, everyone says eat more fiber, I did it, it's great, but you don't know if it's really the method or it's because of just their workflow is changing. So I do want to point out, it's not all bad news. This is comparing two different methods. One method is performing a lot more consistently. This is what we go forward with now. But these are really challenges I want to bring to you, because these are things my lab's working on now, but it's going to take more than just us. So how do we, when we think about designing these waste management systems, I really wanted to say we've solved it, right? We've solved this problem. But I would really say we're really at the beginning of understanding how to measure the problem. We're not at the ability of saying, this is what you should do, other than the cleaner, the more treatment mechanisms you have, the safer your water is. But I think overall, I frame this for environmental metagenomics, but we have to think about how do we overcome the lack of comparability in metagenomic methods in general. So this could be in the human microbiome space, in the environmental microbiome space. We have very similar challenges. But we're all trying to address them kind of in different ways. So that's really all I have. So great, I hope you have questions.
Questions?
So here in California, the water quality at our beaches is a big issue, and it's fecal bacteria that get measured, and entities like Surfrider do that sampling, and they do a lot less of it than I expected, and I think it's because of this measurement, the ability to measure, and they do it in their own lab. And the state of California says, oh, thank you very much for those readings, but it's not posted anywhere for our beaches. And I know this is aside from city sewers, but if you could comment on that, and if there's any thought of doing better with that, but I'm sure it plays into this lab variability.
Yeah, so I would say when I think about environmental microbiology, I'm always trying to compare to clinical microbiology because they have figured it out. I also think they have a slightly more consistent system in general because humans are at one temperature, whereas beaches and environmental microbes are at very different temperatures. But yeah, so I think in culturing, we are fairly consistent because the EPA has published standardized methods. However, for these sequencing technologies, they've only been around, I would say, since 2011 or 12 is when they really got going. So we're still we're still only 10 years into it. But work at NIST and work my lab is doing is developing things like internal standards or different ways that you can turn your comparative data into quantitative data that actually really lets you characterize the bias in your sample, because every workflow is going to have biases. That's okay, because you have different things you want to target. But if we can quantify that bias, it might make things more comparable. So if you could have 2 different labs where you say one knows they're gonna lose something, and that's okay, they don't really care about it, we can start to move forward in being a little bit more open with how we talk about this. Yeah.
Hello?
Yeah.
Okay. Hi, I'm Suki Yem from Cabrera High School. I'm a junior, and I wanted to focus more on the waste management aspect. I read an article concerning California about the waste being dumped into the oceans, and I just wanted to know your comments on the ideations of the spread of diseases to like our ecosystems in the ocean, as well as will it be different in other parts of our country or our world because of the different oceans and different life forms that take that?
Yeah, so I think you get at a lot of different questions, which are super, super great and things we like to think about. So yeah, we talked about here about specifically sewage systems, but we are really worried about things like stormwater runoff. I think right now in California, you're thinking about this a lot, but when you get these big flooding events, they cause a lot of runoff into surface waters and into different places. So this is an issue everywhere. Stormwater runoff is a huge challenge. And we are, specifically for antibiotic resistance, we are worried about it getting into the environment because increasingly we're finding that clinical isolates can actually survive in environmental waterways. And we worry about it, I would say, in the environmental engineering space, we're pretty human-centric. So we're worried about it in terms of human risk, like if you were surfing and there was resistant bacteria. But we are also worried about kind of the ecosystem risk of some of these, we call them kind of like invasive microbial communities, that they weren't natively there. We have a challenge in how to measure that and how to understand what's normal and what's a problem. I don't think we have kind of that idea of what does a healthy ocean microbiome look like versus a problematic one. Well, we do for a little bit for algae blooms, but nothing really more elegant than that. So I think people are thinking about this, and also can more resistance evolve? But it is still an active area of research. We do a lot of monitoring for fecal bacteria, but we can do better in terms of more bacteria that we could be looking at.
I'm Helen from Samueli Academy. I'm a junior there. And I had a question about you said you mentioned the recovery of bacteria in the distribution phases. And I was wondering if you foresee a change in the distribution process to account for that or accommodate that, or if that's just kind of the norm and that will keep.
Yeah, so I think it depends on the system. So in a lot of the non-potable systems, they are supposed to be maintaining a chlorine residual, which is going to keep that microbial regrowth. But because reuse water is inherently, it's not clean, it's not drinking water, we are piping it around, we're distributing it in a way that we haven't really thought about having chlorine residuals. We're seeing this regrowth, so I do think utilities are noticing this and they're thinking about what they can do. Because chlorine and chloramine is what we've always used for drinking water. And so the idea was like, oh, we'll use it for reuse water. That'll be great. However, they have really different water chemistries, which means there's different kinds of bacteria there. So I think people are noticing. We do know for things like potable reuse that you can get your water to a very safe level. You can get it to drinking water standards. It's just a lot more expensive. And really for reuse, people want the nutrients left in the water. That's one of the advantages, is you need less fertilizer, but it's this trade-off. So maybe it really is giving advice on how you should distribute that water. So making sure people know, like I said, don't use aerosolizing sprinklers, use drip irrigation. So things that aren't gonna present risks to like occupational health or residential health. So I think there are always ways we can make the system work, but really that communication piece and trying to understand what should we be looking at here.
Hi, I'm Lucas Ortega from Samueli Academy, and you talked about how humanity is starting to run out of fresh water. And I was wondering, with a rising population, do you think humanity will at one point need to start desalinating ocean water? And if so, what kind of environmental impacts would that have?
Yeah, so I don't think we need to wait. Desalination is here and it's happening around the globe, including in Southern California, but also in places like Singapore and Korea. So we are doing desalination at scale in a lot of places that are water scarce. I think there are a lot of environmental implications of this, especially what do we do with that salt? I think historically the approach was like return it to the ocean. But that just means you're dumping a lot of concentrated salts back into a place and you're increasing that salinity. That can be problematic for marine life. And also that salt can contain a lot of concentrated contaminants that you weren't necessarily meaning to dump in at scale. So I think it's, again, my answer is really always that is manageable, but we need to think about what we're doing with it. I don't think the status quo of dumping the salt back into the environment is probably the solution. We do know that that causes problems for aquatic life, but then it's how do we handle that salt? I know a lot of people have said, oh, well, you can buy sea salt, why don't we use that? You don't want to eat the salt. This is not good salt. But yeah, I think we do need to think about what we do with it and where we discharge it. But we are addressing the desalination challenge or the water crisis. But it's really expensive. So I think, again, we always come back to, let's not waste water where we don't need to. But if we have to, it's hugely energy intensive to desalinate, but it is a solution that we have. Oh, sorry, I'll keep going for 1 second. That really works if you're on a coast, but if you're not on a coast, that can be, you don't have any water to pull from. And so especially when we think about arid regions like New Mexico and Arizona, water reuse becomes a much more viable option because there's no water there. Yeah.
So maybe we're here.
Hi, my name is Walter Sevilla. I'm from Samueli Academy. Sorry, I stepped on my thing. Your presentation had a lot of graphs and data, and I was kind of struggling to interpret it. Could you refer me to some resources on how to better understand it?
Yeah, so I can refer you to, I will be honest, we don't have great ways to educate people on how to do this. This is part of what I do in my lab. But I think it's really starting with kind of honestly biology and understanding how we can culture and grow things just from biological science in general, honestly, before we get to the engineering. I wish I was at a stage where I was designing treatment processes. That's what I thought I was going to do when I started grad school, but we're still at the stage of diagnosing the problem. But yeah, so honestly, I think it's starting with really more microbiology textbooks on how do we culture things and how do we interpret culture data, and then moving into things like next generation sequencing challenge. So this is a big area of where we're realizing that students don't ever get educated on how to do this stuff, so we're working on it on the academic side to try to bring that into curriculums at like the high school level, because increasingly all like health sciences and environmental sciences are using it.
Is there a question over there? Right here.
The variability in this way.
I can hear you. I'll repeat it if we need to. Okay.
The variability that you saw across the labs, I have experienced that as well with a particular cancer biomarker. But when we stuck with the same lab, we saw a good consistency to the point that it was reliable to track the patient, and the variability had a lot to do with instrumentation and protocols. So I'm just wondering whether you did any experiments to really look at the variability in the same lab as opposed to across the laboratories.
Yeah, so actually most of the studies that are done, we can get those really tight error bars and that really low variability when it's one lab. So that we're confident really that an individual paper's results are true. The question is when we start to aggregate, how do we build on that knowledge? So yeah, I think at the individual scale, we're finding that things are reproducible. I think a lot of the applications or things that we want to do from this antibiotic resistance monitoring kind of at scale is to look at global distribution of resistance genes and kind of their emergence and transmission. And that requires us to have a lot of labs that are generating data that's comparable. And I guess similarly, things like public health surveillance, where we're measuring things like SARS-CoV-2 or other public health indicators, we have to have a network of labs kind of all conducting things, and it's really, yeah, so that's where the comparability kind of at the national scale becomes really important. But I do think individual labs can show that they are doing really reproducible work.
Good. Any other questions? Okay, thank you.
All right.
Ishi.